Welcome to AP Biology. Today's presentation is going to be on translation. So we have our messenger RNA code, and now we need to translate it into a protein code. So this is going to take place at the ribosomes, and the messenger RNA is going to go out, and the MG cap on the messenger RNA is going to help it attach to a ribosome, and then the two ribosomal subunits are going to come together. And they're going to start scanning the messenger RNA looking for the start codon. So the start codon is AUG. And when they find a start codon, they will stop when the start codon is in the P site, and they will signal transfer RNAs to bring in the appropriate match to the start codon. Now, it's not really a true match, it's the complement. So AUG, which is our codon, is going to be complemented by UAC, which is what we call the anticodon. So you have an anticodon on your transfer RNA that is the complement to your messenger RNA. Complement means the one that would match up with it. So A with U, A with U, G with C. And also on the other end of that transfer RNA is going to be an amino acid. Okay? We call these the E, the P, and the A site. The A site is the attachment site. The P site is where the growing polypeptide is attached. And the E site is the exit site. So once this is attached, then another signal will go out to bring in the next transfer RNA. So the next transfer RNA will come in, and it will bind right here, and its anticodon would be GCG, because it's going to complement this, and it's going to carry in arginine, and then a peptide bond would form between this methionine and the arginine, and then it would slide down one more, and now this amino acid is going to have the growing polypeptide chain attached to it. This original amino acid would be released because it's in the exit site, and a new one would come in and attach. And that would just continue till the protein is made. So here you can see it kind of in a cyclical fashion. Okay, so you have a new one coming into the attachment site. You get a bond that forms between the two amino acids. Then everything slides over one, and then this amino acid or this transfer RNA will be released from the exit site, and then a new amino acid will come back into the attachment site, and a new bond will form, and then slides over one, and this cycle just keeps going and going and going until you have your polypeptide, which eventually will be your protein completed. When you get to the very end, okay, there will be a stop codon. A stop codon is going to code for a releasing factor. It's going to come in and bind. There will be nothing more for the polypeptide growing chain to attach to, and so it just gets released, and then that released chain is like a free polypeptide. It's often in the ER, but sometimes it's just in the cytoplasm, and it usually has to get processed before it will become functional. Lots of amino acids can be combined on different ribosomes all at the same time. So because this piece of messenger RNA is not going to last very long, because we said, remember, the exonucleases are eating it from the ends, so it's not going to last very long. So as soon as one ribosome attaches and starts making a protein, another one will attach right behind it, another one right behind that, another one right behind that. And there's not hardly any space between them. They're just making their way along the messenger RNA, trying to make the little polypeptide chains that will eventually become proteins. You can see it here in a bacteria. These are the ribosomes attached to a messenger RNA. So you can see they're pretty crowded on there, trying to get proteins made as quickly as possible. So once the protein or the polypeptide chain is initially made, it usually has to be processed. A lot of times they're made directly into the ER, and then they'll be shipped from the ER to the Golgi in the vesicle, and then in the Golgi they get all their final processing. So what do we mean by processing? Well, the Golgi will help them fold appropriately. Sometimes you have to add a sugar to a protein, and you end up forming what's called a glycoprotein because you're attaching sugars to proteins. Sometimes you will add a fat to a protein, and you end up forming what's called a lipoprotein. But yeah, there's lots of modifications that take place to proteins before they actually become functional inside the cell or at some other cell.